Hi there. In this video, I'll be talking about the ways that DNA is segregated among the daughter cells during cell division. This includes some of the segregation called fission in both bacteria and some eukaryotes, as well as mitosis and meiosis. This is a two-part series of videos. This first part will go over some of the basics, and then in the second part, in a different video, I'll talk about some more unusual types of segregation of DNA. So one of the simplest forms of segregation of DNA is fission, as you see in bacteria and archaea, although some eukaryotes also divide their DNA by a fission-like process. As indicated here, the DNA is not just randomly organized within these cells. There's a very definite pattern of organization, including the attachment of some proteins to the membrane and other proteins that are attached to the membrane. During initiation of cell division, the DNA begins to replicate, but the number of proteins attaching the DNA to the membranes also increases, so each one of those new chromosomes has an attachment site to the membrane. As the process continues, the cell also grows and lengthens, and part of the process is that these chromosomes are then beginning to be pulled to opposite poles of the mother cell in preparation for cell division. The segregation of the two replicated DNA molecules then is complete when they reach the periphery, the outer periphery of those cells. And then in the middle of the cells, the membrane and cell walls begin to invaginate to begin the separation of the two cells. Finally, the two cells are divided by the extension of the membrane and cell wall material. Although if foreign DNA is to enter those cells, normally it enters through those softer parts of each of one of those cells in between as they're dividing. The cell wall material isn't quite completely built up in those areas. Then this process can repeat over and over again while those cells in the population are dividing. So the cells can also stop dividing depending on nutrition, temperature, and other factors. But this process can repeat over and over and over again. And in E. coli, this process entirely occurs within about 20 to 30 minutes under optimal conditions. There actually are some additional things that can happen to replicate and divide the DNA. So this one here is just a schematic of what I was just talking about, binary fission. You start out with a 1C level of DNA in the bacterial cell that duplicates, so now you have a cell that's actually at the 2C level. Then you get fission, and you have two bacteria, each at the 1C level. So that's the common way to undergo cell division, replication and cell division. However, some bacteria undergo replication without having cell division, so they become polyploid. They have many copies of their chromosomes. So many types of bacteria are capable of this, and they normally do this process when they need multiple copies of certain genes, not necessarily the entire genome, but especially certain genes like ribosomal gene copies and things like that. They can replicate the DNA so they have more of those genes, more copies of those genes, so they can produce more of those RNAs or proteins, whatever is needed in the particular case. So it's actually quite common. Another process that occurs, especially when bacteria are under stress, that is, that they're lacking some nutrient from the outside. And so they need to form some cells that will actually produce that nutrient or somehow save them from perishing. And so they can actually undergo extra chromosomal amplification, and they can also reduce the number of repair enzymes that they have. So the DNA is going to mutate at a more rapid rate than normal. So they will form small circles of DNA with certain genes on them, and probably a random process. And during this replication process, there are mutations that occur, and they're not repaired because they've reduced their repair systems, so that they have many circles having mutations of particular genes. The cells that are particularly successful, that is, they start to grow again in that particular environment, then reinsert those genes into the proper place in the genome, and then go on to form colonies that survive that stress, whatever it was. So when this was first discovered, people didn't know actually what the mechanism was, and it looked more like a Lamarckian type of evolutionary process where the environment was causing mutations that would be beneficial for that organism to survive in that environment. What was found was that the organisms reduced their repair system activities, and this actually increased the mutation rate. So the environment really was not causing the mutations, it was just signaling the bacteria to stop repairing all of its mutations. And this phenomenon was first discovered when some students or someone working in the lab left some plates over an extended period of time. So these were selective plates. These were plates that were lacking a particular amino acid. And so the organisms, the bacteria, would only grow on them if they had a mutation. When they came back to the lab, they found that the number of colonies on those plates was far greater than what would be expected just by simple mutation. So it appeared that the bacteria were evolving, essentially, to grow on that particular medium that was missing an amino acid. And originally the bacteria that were put into those culture plates lacked the ability to produce that particular amino acid. So they were that amino acid minus, and the medium didn't have that amino acid in it. So the only way that they could survive would be to repair the mutant gene, which would then allow them to make that amino acid and survive. So this appeared to be an example of Lamarckian type evolution. So there were actually quite a few papers written from the time the students or the researchers found those increased rates of mutation in those bacteria to the time that someone finally discovered how the process was working, and that it wasn't Lamarckian, it was actually Mendelian, with a reduced repair mechanism, so an increased rate of mutation in those bacterial cells. And then finally, there are extra chromosomal units in bacteria, as well as some eukaryotes called plasmids, which are autonomously replicating sequences. That is, they have an origin of replication, and they can replicate over and over and over again. Some produce only one or a few copies per cell, but then there are some that produce hundreds of copies of these plasmids per cell, and these are actually used for cloning and things like that in laboratories. So that's one other means to replicate certain sequences. Antibiotic resistance, often those genes are encoded on these plasmids, and the plasmids can be shared not only with the members of the same species, but they can also be shared with members of other species. So this is how antibiotic resistance gets around very rapidly in some cases. So each of these can be explained on a typical cell cycle. So here's a cell cycle of bacteria and archaea, where you have cells in the G1 phase, some of which go through a differentiation or a resting stage by forming spores. Then you go into the S phase, where the level of DNA is increased from the 1C to the 2C level. And then in the G2, which is the 2C level, also cells can undergo differentiation or resting stage in a spore, etc. And then through binary fission, where the cells are divided and the two chromosomes are separated to yield a 1C cell again. To get a polyploid cell, you can skip G2 and fission and just continually go through G1 and then S and then G1 again and S and G1 and S again. And in each case, the number of chromosomes is increasing. So you go here to 2C, and the next would be 4C, the next would be 8C, etc. For extra chromosomal amplification, then for some parts of the genome, those pieces are replicated over and over and over again, rather than replicating all of the genome. For plasmids, there would kind of be an offshoot of this, where you would have a separate cell cycle where those pieces would be replicated over and over again. And the number of copies of those plasmids varies from species to species. So if only one copy is produced, then when the cells divide, sometimes one cell gets a few copies of the plasmid and some cells don't. In the case of high copy number plasmids, where you're having the cells will have from 100 to 200 copies, something like that, then both cells receive an appreciable number of those plasmids. And by the way, these lines separating G1 and S and G2, etc., are dependent on specific proteins and enzymes and signaling molecules that will send these cells into each one of those phases. So although we kind of draw lines there, those lines actually represent some signals that are being delivered to those cells. So the cell cycle in eukaryotes is a bit more complex, in that there's this mitosis stage. So you have a G1 phase, where the cells are either at 2C or 1C, depending on the species. And then you have differentiation or apoptosis, which is cell death. They can also go into a resting stage. Then they go into the S phase, where the amount of DNA is doubled, either from a 2C to a 4C level or from a 1C to a 2C level. And then into the G2 phase, where the cells are 4C or 2C, depending on the cell type. And they can also go into a resting stage here, and then into mitosis, where you have prophase, prometaphase, metaphase, anaphase, and telophase. And then finally, this K stands for cytokinesis. The cells divide. And by the way, there are specific signaling molecules that are turned on at these places, these transitions from G1 to S and from S to G2, and then in each one of the phases of mitosis and cytokinesis, etc. Some of these are very important in cancer regulation. So they were actually discovered in yeasts, but they turn out to be fairly important regulators in the progression of cancer. So these lines actually do have some importance, aside from just separating the two phases. There are molecular mechanisms involved. So in most bacteria, the chromosomes are circular, or they undergo that RDR type of replication. In eukaryotes, replication begins at a replication site, an origin of replication site, but they're located at many places along the chromosomes. And in the vast majority of cases, the chromosomes are linear. And so this presents a few problems. As the chromosome replicates, the leading strand can go all the way to the end. The lagging strand, on the other hand, has to produce these Okazaki fragments, starting from a primer and then going through to the next Okazaki fragment, and finally putting those together with ligase. However, then that presents a problem because you can't get all the way out here on the 3' end. So the chromosomes, if they aren't protected on those ends, become shorter and shorter and shorter with each replication cycle. And actually your chromosomes are becoming shorter and shorter and shorter as you age, as your cells divide. But there's a mechanism for protecting parts of the ends, and that's done by using a telomerase to produce telomeres. So these are repetitive regions that are on the ends of your chromosomes, and these enzymes, the telomerases, are active during your early development. And so by this time they've turned off, and your telomeres are usually as long as they're going to get. Telomerases have a reverse transcriptase activity, so you do have reverse transcriptases in your body that were active early in development. And they also carry an RNA primer. And so what the RNA primer does is it recognizes a part of the end of the chromosome and then starts making a new DNA strand attached to the ends of each of the chromosomes. So once it's finished polymerizing DNA on that strand, it then flips down to the next region and polymerizes that further and keeps going down. So then you've got hundreds of these repetitive elements on the ends of your chromosomes. But it only produces one strand. So how do you get that other strand and make a double-stranded telomere? So that's filled in then by DNA polymerase. So at the end, after the telomere has added those ends on, then the DNA folds over on the end, you get DNA polymerase, then filling in the other side of that chromosome, the other strand of the chromosome. So now you have a long telomere consisting of those telomeric repeats on the ends of those chromosomes. So each time the DNA, that chromosome, is replicated, it loses a little, but it only loses those repetitive telomere sequences. And at least early in life, you don't lose any of the genes that are further in from the telomere. Later in life, those start getting eaten away, and that's part of the aging process. This is a fluorescence microscope picture where they've made a probe that can hybridize to the telomere regions, and then it'll light up in this whitish color. So you can see on the ends of all these chromosomes, you have telomeres on both ends of all of those chromosomes. So those are metaphase chromosomes that look something like this X up here. But if you start dissecting that away, pulling different proteins off, the first thing you can see is that the chromosome is arranged so that there's a central scaffold made up of proteins and RNAs, actually. And the DNA loops out. And each one of those loops is approximately 300 nanometers in length. And then you can look at a very small region of that and find these 30 nanometer structures in the electron microscope. And these are actually the DNAs wrapped around histones. So you can see that. You can remove histone 1, which is this central one, and then you get what are called beads on a string. So you can actually see these in the electron microscope. And these are the DNAs that are wrapped around the histones, but the H1 histones are not connecting those together. So you've got about 100 and I think 60 nucleotides that are between each one of the histone units, the beads on the string. And by the way, while bacteria don't have histones, they have other positively charged proteins that are histone-like. But archaea have histones and eukaryotes have histones. So these began in earnest in the archaea, and we're related to archaea, so that makes sense. And then if you look at just the DNA itself, it's 2 nanometers across. So here's an electron microscope picture, actually a high voltage electron microscope view of a chromosome. And you can see all these loop outs of these DNAs, but they're also somewhat folded up. And so the histones are folding them up, and then these are being attached to a central scaffold to hold the chromosomes together. And this is a composite electron microscope picture. That's why you can see these kind of joining places where they've joined this together, because they couldn't get one view of this chromosome. And you can see they've taken off the histones and other proteins that bind up the DNA. And so you can see this central scaffold. And again, that's made up of proteins and RNAs. And then all of this out here, this fuzzy area, that's all of the DNA that's attached to those chromosomes, those central scaffolds. And here's a close-up of the scaffold at the bottom, and then all these loops of the DNA, again, without the histone. So these have been sort of spread out so that you can see it. And if you follow each one of those little lines, they go out and then circle back to that central scaffold, and then the next one goes out and circles back. So it's one long piece of a chromosome of DNA. This is just a diagram, more of a stylistic diagram of a chromosome. So, but it does point out a few things. One is that you have this central primary constriction, which is the centromere of the chromosome, where the kinetochores attach and the microtubules attach to pull those chromosomes apart. Often, on many chromosomes, not all, you have a secondary constriction. These are kind of a misnomer in that actually the DNA there is not highly constricted. It's actually looping out, but it doesn't stain well, so you can't see it. So it looks like another constriction. These are usually the places that you find ribosomal gene loci. So many ribosomal genes are in there, and they're constantly transcribing, which is why they're not condensed like the rest of the chromosome. When you stain the chromosomes with different stains, you can also see banding often. So you can do what's called a karyotype of human chromosomes and look at the banding pattern. Sometimes in cancer, then you can determine what's happened, what the fault that's occurred in the particular cancer. So there are G and R bands often. G is for a particular stain called Giemsa, and R are the places that don't stain with Giemsa. So you can see this banding pattern. But again, this is a metaphase chromosome consisting of 2 chromatids, which are each copies of that chromosome. So during mitosis, you see this where there are 2 chromatids attached together at the centromere, which is where the kinetochores attach to. Under the same conditions, just a diploid cell would have 2 homologous chromosomes that, if they were condensed, they would look like this, but they're not normally condensed. But realize that each one of these chromatids is actually a copy of the chromosome. So although this whole thing is called a chromosome, it really consists of 2 copies of this chromosome over here. Now, to copy a bacterial chromosome, normally they have 1 or 2, sometimes more, origins of replication. E. coli has 1. So having 1 replication origin is sufficient to be able to copy the chromosome in less than 20 minutes, which is the doubling time of, or division time of that bacterium. The amount of DNA in a human cell or any eukaryote is multiples, hundreds of times, sometimes thousands of times larger than that of a bacterium. So one origin of replication is woefully insufficient to copy all of that DNA for one cell cycle, which in humans is about once every 24 hours for rapidly dividing cells. So instead, there are multiple origins of replication. So this just shows the long arm of chromosome 21, human chromosome 21, which is one of the smaller chromosomes in the human genome. And so all of these bands in white are pieces of the DNA that are pieces of the chromosome that are replicating early in the S phase. And then the ones in gray are in mid-S phase, and finally close to the centromere, you're having replication, the latest parts of the chromosome being replicated. And other studies have been done on different parts of the human genome, so different chromosomes, the long and short arm, finding the same thing, where you have this various patterns of initiation of replication all over throughout the chromosomes. So then during the cell cycle, you have the DNA unwinding from the histones in order to be replicated. Then in preparation for mitosis or cell division, you have the histones wrapping up around the DNA, those wrapping up further, and then that happens with all 23 pairs of chromosomes in the human genome, or depending on which organism you're talking about, it would be 6 pairs of chromosomes in broadbean and pairs of chromosomes in all the others. So the cell has to be very organized, and the DNA has to be very organized in order to do these processes. So the nucleus is highly organized as to where each one of those chromosomes is in the nucleus. And then when they're replicated and separated so that they're not tangled all together. So that, and then organized eventually for mitosis, so that one set of the chromosomes winds up in each of the 2 daughter cells during cell division. Now, eukaryotes have ranges of chromosomes. The smallest one, of course, has 1 chromosome. That's a certain plant has 1 chromosome. And up to 630, although I think there are some that have over 1,000 chromosomes, and that's per haploid set. So in this one, you would have over 1,260 chromosomes per nucleus, and you have to make sure that that the cell replicates those, keeps them separate, and then during mitosis equally divides those between the 2 daughter cells. So here's just an overview of how that occurs. You have a cell at interphase at G1, and then after the S phase, you have a cell at G2. Note that it has a larger nucleus because it's doubled the amount of DNA, and it has 2 nucleoli, which is where the ribosomal RNAs are transcribed, and the ribosome subunits, the small, large subunits of the ribosome, are put together. In prophase, you can notice that the DNA is starting to condense. So proteins are being added to the DNA to condense those. And then there's a prometaphase where the chromosomes are already condensed fully, but the microtubules are being formed. So that's prometaphase. And these microtubules continue to grow and attach to the chromosomes, and they will push the chromosomes out to the metaphase plate, the middle of the cell, usually, not always in all species. And then a separate set of microtubules from the opposite poles will attach to the kinetochore, or the centromere region, from the other pole of microtubules. And there are actually 2 sets of microtubules in here. Ones that are called kinetochore microtubules that attach to the centromeres of the chromosomes. And then there are polar microtubules that cross each other in the center of the cell, and they do 2 very different things. And you can see that here. There's anaphase, where the the chromosomes are being pulled by the microtubules, as well as proteins called dyneins, being pulled to the poles. But then those polar microtubules are still crossed in the middle of the cell, and you can see the cell elongating, because those microtubules are actually growing, and they are pushing the two poles of the cell apart using a different type of motor protein called kinesins. And then finally, in the center of the cell, the cytosol and the membranes are pulled together using actin molecules. And that's in animal cells and protist cells. Plant cells do it a little differently, and I'll talk about that later. But this would be for animal cells. And finally, you get cytokinesis, division of the two cells. This diagram shows just a few of the molecules that are involved in this process. So these have been stained specifically with fluorescent dyes that attach to specific molecules in the cell. You can see the Golgi. The Golgi are responsible for modifying certain proteins and that are, in this case, important to production of the microtubules and actins. And so here you see beta-tubulin, that there's a microtubule spindle that is pulling apart the chromosomes, moving them to the poles, and they're being squeezed together here by actins, which aren't shown in this particular picture. And then you see the DNA stained in a different color, and then there's the composite of all three. But these aren't the only molecules that are responsible for cell mitosis and cell division, but just to show you that a lot of different things are happening at the same time during this part of the cell cycle. So in this diagram, I just wanted to illustrate how the chromosomes are being replicated and then separated during mitosis. I'll do the same thing for meiosis in a later video. So here are the two homologous chromosomes in a diploid. They, of course, wouldn't be condensed like this, but I just wanted to show them diagrammatically in this case. So during the S phase, you get duplication of each one of those. So now you're at a 4C level. In the case of a diploid organism, go from 2C to 4C. These are called sister chromatids, right? In this case, although they're duplications of those homologous chromosomes. And then during mitosis, these get attached to get attached to microtubules through their kinetochores at the centromere. And then the sister chromatids are pulled apart during mitosis, and thus becoming separate chromosomes. So the terminology changes from a chromatid to a chromosome in that case. And you wind up with now two nuclei, or two cells, each with a pair of those homologous chromosomes again. If this were a haploid cell, then you would just start with one of these chromosomes, build up to two chromatids, separate those, and then wind up with a haploid set of nuclei as well, two of them. There are two basic types of mitosis, one called astral mitosis, and aster looks like this, which contains a centriole, and that's where the microtubules are arranged. So it's called also a microtubule organizing center. And aster just means star, so it looks like a star pattern. And then there's anastromitosis, where you don't have a centriole, but you still have a microtubule organizing center, but it's more diffuse. And this occurs in plants and some other organisms. But in animals, you would see this astromitosis up here, where the kinetochore microtubules attached to the centromere of the chromosomes would be pulling the chromosomes to each pole, and the polar microtubules would be crossing in the center to stretch out the cell, to expand the cell. In the case of anastromitosis, you would then have a more diffuse area, but still a microtubule organizing center, the kinetochore microtubules, and the polar microtubules would emanate from those. Still, the chromosomes would be pulled by their kinetochores or centromeres to the poles, and these microtubules crossing in the center of the polar microtubules would be pushing the cell apart, or at least pushing the microtubule organizing centers apart. Now, in both of these cases, I've also shown that the nuclear membrane is disintegrating, and I'll show you many cases where it does not disintegrate, and some other special cases too. There are some plants and some animals also that have holocentric chromosomes, so they don't have a centromere, but they're attached along their entire length to the microtubules by kinetochore-like proteins. So they're pulled pretty much in parallel to one another, apart to each of the poles of the cell. There also are many species of organisms that have a nuclear membrane that never breaks down during mitosis. So in most dinoflagellates, which are in the alveolata group, they have the chromosomes within a nucleus, and often you can't see them like this. And the microtubules pass through tunnels, so the nuclear membrane is fully intact, but it forms these little tunnels through where the microtubules pass through. During mitosis, then, these microtubules act to pull the nucleus and the attached chromosomes apart equally, one set to each new cell. And if you think about it, this resembles fission, the separation of the chromosomes in bacteria and archaea, where the cell membrane in this case does not break down. You just pull these things apart by attachments to the cell membrane. And that's similar to what's happening in dinoflagellates, but instead of this being the cell, now this is the nucleus of the cell. But it looks very similar to what happens in a bacterium or archaea. In some dinoflagellates, they do have a centriole and an aster. And in this case also, they do have true polar microtubules and kinetochore microtubules. But the chromosomes are attached through the membrane, through these proteins, to the microtubules. And then when those separate, these microtubules essentially pull the nuclear membrane apart and form two new nuclei on both ends of the cell with one set of each of the chromosomes. In Ascomycetes fungi, so these are a certain group of fungi, the microtubules form within the nucleus. And so you have a set that resemble polar nuclei and a set that are like kinetochore microtubules. There also are other microtubules outside the nucleus that are pulling on the nucleus as well. So these aren't the only ones that are formed. So when these separate, then it pretty much makes a dumbbell shape out of the nucleus, similar to what you have in dinoflagellates, except the microtubules now are inside, fully inside the nucleus. In basidiomycete fungi, the chromosomes are actually ripped out of the original nucleus. And then as these chromosomes begin to move to the poles of those cells, new nuclear membranes are constructed or built up around those chromosomes. So it's quite different from the Ascomycete version, where that nucleus never disintegrates. Here, the nucleus disintegrates in one end of the cell after those chromosomes have been pulled out of it. So here are a couple of schematic diagrams of that. Ascomycetes on the top and basidiomycetes on the bottom. So here's the Ascomycetes, and the chromosomes, you never can see individual chromosomes. It's just a dark staining blob on one end of the cell, or one end of the nucleus. The first thing that happens is a set of microtubules starts pushing out this bud, and that's why they're called budding yeast, and they divide by fission. And then the chromosomes duplicate, they're moved to opposite poles of the nucleus, and then you have other microtubules outside of the nucleus that are attaching that nucleus to these other membranes. The chromosomes begin to be separated by growth of the microtubules. Eventually you get splitting of the two nuclei with one set of chromosomes in each case. And finally, cell division in this case, which is a budding form of fission. For basidiomycetes, it's a little more bizarre in that here's the chromosome again, you have duplication of the chromosome, and then extension of these microtubules outside of the nucleus. And those pull on those chromosomes, essentially ripping them out of the nucleus. You then start getting growth of those microtubules and then attachment to the opposite membrane, and the chromosomes begin to separate. And as they're separating, new nuclear membrane forms around each of those sets of chromosomes. And you can see in one corner of the cell that nuclear membrane breaking down, and some of those pieces are probably adding to the new membrane. And then finally, you have cell division or cytokinesis at the end. So I want to describe a few more details about anaphase, because the polar and the kinetochore microtubules are doing two different things at the same time. Anaphase can be split into two parts, anaphase A and anaphase B, and they're essentially occurring simultaneously. So anaphase A consists of the chromosomes being pulled to the ends of the cells, to the poles of the cells, by the kinetochore microtubules, which have attached to them a motor protein called dynein. So the dynein is walking along the microtubule as the microtubules are being disassembled. The other set of microtubules, the ones that are pushing the poles apart, are doing something very different. They're attached to different motor proteins called kinesins, which also walk along those microtubules. And you're getting addition of tubulin to the ends of those microtubules to lengthen the microtubules. So this set of microtubules is lengthening, and the set of microtubules that are pulling the chromosomes apart are shortening. So this shows it in a little bit more diagrammatic form. Here are the polar microtubules here, lengthening on their plus ends. They have a plus and a minus end. And the kinetochore microtubules are shortening from their plus end and pulling those chromatids, now chromosomes after they separate, to the two poles equally. So there are many kinds of motor proteins that move along microtubules called MAPs, microtubule associated proteins. One set are the dyneins and another set are the kinesins. And the dyneins move towards the minus end of the microtubule, and the kinesins walk towards or move towards the plus end of the microtubule. And they all occur as dimers of these proteins, where one foot or head is actually on the microtubule at all time, and then the other one moves and attaches to the microtubule, the other one lifts up and moves. So they actually have a walking form. They look like they're walking. The other ends are attached to cargo, whether it's a vesicle or a chromosome or whatever. And both work by hydrolyzing ATP for those movements. When certain things are measured in a cell during mitosis, you can see some of these things occurring. So initially during prophase and prometaphase, the amount of GTPase is increasing, which causes the formation of more GTP. GTP is needed for the polymerization of microtubules, so that goes up, and so you see during prometaphase and metaphase an increase in the number of microtubules, which is the case. Later on, you see the GTPase and GTP levels dropping down, and then the calcium levels coming up, and calcium is used sometimes for depolymerization of microtubules. So you see part of that, but that's only with the kinetochore microtubules, which are then pulling the chromosomes to the poles. But you also see about the same time another increase in GTPase and an increase in GTP, which is causing the polymerization of the polar microtubules. So eventually all those go down by the G1 phase and cytokinesis, which will separate the two cells. And one other thing about the disintegration of the nucleus, it doesn't happen in all cells, but in a large number of cells, the nucleus disintegrates or seems to go away, but it doesn't actually completely go away. There are certain proteins called lamins, which are intermediate filaments, that essentially form the nucleus. And so they look like this in an electron micrograph, and so they look sort of like burlap or fabric. And on one side of those, they attach the nuclear membrane components. And on the other side, they will attach DNA. So they really are the thing that forms the nucleus. And they are sensitive to phosphorylation. So when those proteins are phosphorylated, they tend to dissociate from the chromosomes. And then, but they are still hanging on to bits and pieces of the membrane. During early telophase, when the nucleus is reforming, they are dephosphorylated. Once they're dephosphorylated, they start associating together, bringing those pieces of the membrane back together. And the dephosphorylation also allows them to attach to the chromosomes again. So then essentially the lamins are forming around the chromosomes, and they're bringing membranes with them to reform the nucleus around the nuclear membrane around the chromosomes, with inclusion of the nuclear pore structures as well. And before I finish, I just want to mention the differences that you see in plant cell division. Plants have a rigid cell wall around them, so that limits whether you can stretch this cell and have things divided in exactly the same way. Plus, you can't divide the cell just with membrane. You have to build up cell wall material between the two new cells. So on top here, these are light microscope pictures using a special feature that makes them look sort of 3D. And actually a friend of mine back in graduate school did these a long time ago. Had to spend multiple hours in a cold room with a microscope taking individual photos. It can probably be done in a better way today. But here's the interpretation down here, where you have prophase here, where the chromosomes are beginning to condense and the nuclear membrane is breaking down, or at least dissociating. And prometaphase, where you have the microtubules beginning to form. Metaphase, where now the chromosomes are lined up in the middle of the cell. Anaphase, they're being separated from one pole to the other. That is the chromosomes, which were chromatids over here. And the polar microtubules are there also, pushing those polar regions up against the far sides of those cells. The nuclear membranes then begin to form around each set of chromosomes, And a new set of microtubules appears in the middle of the cell. So these are completely different than the polar microtubules and the kinetochore microtubules. These are new microtubules set up in the middle of the cell, which will bring cellulose synthase enzymes to that region to build up new cell wall material, so that eventually you have a new, what's called a cell plate or a new cell wall built up. As it's being built up, it's also moving the membranes around, so then you get two completely separated cells, except they are connected through plasmodesmata between those cell walls. So again, those are completely different microtubules that are being polymerized to build up that new cell wall. Cell walls are also built up around the other parts of the cell during normal cell division by microtubules that are pressed against the sides of those cells to then orient cellulose synthase molecules that extrude the cell wall material outside of the membrane to build up new cell wall material around the entire cell. So again, here is sort of some of the overall general characteristics of mitosis with the polar microtubules and the kinetochore microtubules, both astral and astral, with breakdown of the nuclear membrane caused by phosphorylation of the lamins. But you also have other situations where the membrane does not break down, such as in dinoflagellates and some fungi, etc. And in the next video, I'll talk about some of the possible ways that these different patterns evolved. Okay, so that's the end of this video, which is part 1 of DNA segregation. And as I said, I'll talk a bit more about this in part 2 and include sections on how these evolved, or at least possible ways that each one of these different modes of segregation of chromosomes has occurred. So thanks for listening and watching. I will talk to you later.